monitor. Spore formation meaning method of asexual reproduction. Which are the ones done currently? Asexual methods of reproduction. Which are the ones done then?
Ma'am, fragmentation का भी कर रहा है.
ओके बोला मैंने सब सेपरेट होके बैठो अलग अलग बैठो एक इधर आ जाओ कम एक साथ बैठना ही नहीं अलग अलग बैठो so that you study so that you learn. Now we were talking about the asexual methods. What were the asexual methods? We had already completed with binary and multiple fission. After binary and multiple fission, what next? Fragmentation. We are done with fragmentation. Then after fragmentation, we are done with what was the third? Regeneration. Are we done? No, budding. Are we done? Budding. After fragmentation, what is that method? Spore formation. Okay. So now we continue again with this asexual mode of reproduction. Asexual mode of reproduction. So we continue with this. For asexual mode of reproduction, we had already discussed about fission. There are two types, examples. Next is we had already discussed about the concept of fragmentation. Third is spore formation. Can you give an example of an organism which reproduces by spores? Rhizopus, bread mold, all the different type of fungus would generally show method of reproduction by spore formation, fungus. Now next is we talk about budding. Budding is a method of asexual reproduction. Again, budding we come across in plants. Budding also we come across in some simple organisms, multicellular organisms. So first of all, if I talk about the process of budding, first is I look into certain type of plants. In certain type of plants, example like the bryophyllum. Certain type of plants like the bryophyllum, they show methods of reproduction by budding. Now in these group of organisms, what would happen? The plants, if I talk about bryophyllum, these are short plants and they have got broad, well-developed leaves. The leaves are quite close to the soil. This is not a very tall plant as I mentioned. This is not a very tall plant as I mentioned, and one special feature is they result in the formation of, now that the leaves are all mature, they result in the formation of small buds. Where at the edges of the leaf, they result in the formation of small buds, and from the buds, fine root-like structures develop. As these root-like structures would develop and come in contact with the soil, now these root-like structures would get established in the soil, and after some time, what happens? After a particular period of time, a week or a couple of weeks, what will happen? These leaves will all perish, and then the leaf is all dried out, fall off, perished away. And then what will happen? Whatever rootlets have, come in contact. Now these rootlets have come in contact, so they have already made their attachment with the soil. And after a particular period of time, what are we going to get? Again, a new plant would develop from here, and again a new leaf develops from here. This is the method of asexual reproduction, that is budding, in case of bryophyllum. Again, bryophyllum is one type of a plant to understand process of budding. Let us now talk about budding in some other type of organism like the yeast. The yeast is a fungus and it is a single-celled organism. Again, I am talking about budding as the method of reproduction, asexual method of reproduction. Now here what? This is a single-celled fungus. Fungus can be single-celled organisms and multicellular also. Now that we have been talking about spore formation, there also we were talking about fungus. Which example? Rhizopus. That's a multicellular fungus. Here I am talking about a unicellular fungus. Now this parent yeast will result in the formation of a small bud. After some time, this bud will start growing. If I now take the next stage, this would result in the process of formation of a young organism. So this small bud resulted, or this small bud underwent the process of growth. What about this nucleus? This nucleus underwent division, and now the nuclear components or the nuclei are spread out. Divided. Exactly same like binary fission almost. The nucleus is dividing just once. Then, now that this new organism is formed, is it going to get detached from the parent? Not immediately. This will again result in the formation of another bud like this. And this goes on. And this goes on. So if I have to draw a chain of diagrams, I can just try to summarize in one, mentioning that this type of a structure is generally what we get to see in case of yeast. And what do we call this as? A chain of buds. Where this was the parent organism, this gave rise to a new organism, this will again give rise to a new organism, and this goes on. So these new organisms, they do not immediately detach from the parent, but remain all connected, resulting in the formation of a chain of buds. Now the question is, will they ever detach? Yes, when the chains are too long, the filaments are too long, they abruptly detach. There is no specific number. It is not that only once it is 20, then they will detach. Nothing like that. Just like fragmentation, just random. So what is this? This is known as chain of buds. So I am talking about budding in the yeast. I am talking about budding in bryophyllum. Let us again talk about budding in another type of an organism, and that is Hydra. Budding in Hydra. Hydra is what? It is a multicellular organism. Simple, but a multicellular organism still. So if I talk about this organism, this particular organism, the Hydra that we understand, this is a small cylindrata. It is a invertebrate actually. And this cylindrate has got a structure something like this. It is a slender body. So I would say this is the parent Hydra. What is Hydra? What is its habitat? Hydra is an invertebrate, simple, multicellular organism. Where is it found? It is generally found in the pond, the lakes, shallow water bodies, not generally in the sea and the ocean, and freshwater bodies generally. So this parent Hydra, which is a fine, thin, stick-like organism, slender organism, this will start resulting in the formation of a bud-like structure like this. Now this is the parent organism. It generally has got some finger-like projections. What are these finger-like projections known as? These are known as tentacles. Now these tentacles have nothing to do, since I am talking about reproduction now. The tentacles have some other function, nothing to do with reproduction. But now to understand, what about the process of budding? How will budding happen? Budding would generally happen like this, where this parent organism may develop, again, random number of buds. There is no specific number, that 2 or 10 or 20, nothing. Just at random, few buds will be formed. So this hydra would form few buds. What now next? This hydra will start resulting in the formation of buds, and these buds now will start completing their process of growth. They will continue their process of growth and remaining in contact with the parent body. They will continue their process of growth, remaining in contact with the parent body. Is this new organism getting immediately detached? No, unless and until the growth is complete, they remain in contact with the parent body. Then last is what? The next stage, if I draw, this is the parent hydra. And once the organism is all developed, we come across that these are the daughter hydra organisms, depending on how many buds are there. Depending on how many buds are there. So I would say these are the daughter organisms. Now, can you tell me one major difference between this budding in hydra and fragmentation in spirogyra? What would be one difference? Budding in hydra and binary fission in amoeba. What would be one difference? Check here. This is what you have to understand. What did I draw here? From here, what are you understanding? The new organisms are there along with the parent organism. The parent organism is still existing. Was this the same when we were talking about spirogyra? No, the spirogyra, the entire body was dividing into few fragments, and then every fragment dividing, depending on the length of the fragment, dividing into new organisms. So it was not that the new organisms and the parent organism is parallelly existing. No. What was that term I asked you? What was the point I asked you to remember and write? Here I would say, what is this feature to be mentioned as? I would say, here you are supposed to write as a point of difference, parent identity is maintained. Parent identity is there. Meaning, when the new organisms have already come up, the parents are still existing. But if I talk about the binary fission, if I talk about binary fission, if I talk about fragmentation, what did we write at the time? Parent identity is lost. The parents and the new organisms never survive together. Is this clear? Points of difference is very simple, but see that you pick up absolute marks here, not to lose any marks. These are very, very simple concepts. So this is about budding. So what are the different methods we had come across? We had come across fission, fragmentation, spore formation, budding. Now something different that we are going to talk about would be regeneration. We talk about regeneration. Now is regeneration the same method, or is regeneration to be categorized exactly along with these methods? I would say no. Regeneration we cannot actually categorize along with these methods, because regeneration is a method which is seen in organisms, to only replace the lost parts. That is not a regular method of reproduction. So is regeneration a method of reproduction, a regular method of reproduction? No. It is a method to replace the lost parts. Method to repair the lost parts. Now let us try understanding what does this mean. Replace the lost parts, meaning I would give examples of certain organisms like, say suppose the starfish, say suppose the lizard, the house lizard, say suppose the frogs, and some simple organisms, say suppose the planaria. Now, what are the differences? Try understanding. When we are talking about simple organisms like planaria, say suppose I talk about simple organisms like the planaria. Now, this organism, that is the planaria, what will happen? This organism which is present where again in the freshwater bodies. Planaria again is a platyhelminth. Planaria is a worm. It is a worm-like organism belonging to the group Platyhelminth. Now, this planaria that we talk about, accidentally may divide. Accidentally, its body may be cut into certain fragments. In the case of fragmentation, what did we learn was the body of the Spirogyra naturally divides. The body of the Spirogyra, the thallus of the Spirogyra, the ribbon-like structure of the Spirogyra naturally divides into few fragments. Am I talking about this now? No, I'm saying why does the body divide into certain fragments? Only when the organism comes across certain accidents. Only accidentally the organisms may divide or cut down its body into few fragments. Why accidentally? What does that mean? Quite a number of times, these are small, simple organisms present in the water bodies. Bigger organisms may predate on them. Bigger organisms may eat away parts of their body. That is one thing possible. Number two, how also their body gets divided into few fragments. Quite a number of times, these are simple leaf-like, flat, soft organisms. They come across the shells of harder organisms, and in that process, their body gets torn into pieces. Quite a number of times they come in contact with the weeds and the corals. Their body gets torn into pieces. Quite a number of them they come in between, along with the current of the water. They come in between the rocks and the crevices. Their body gets torn into pieces. So these are all the possibilities why this organism's body would be cut down into pieces. Is it happening naturally? Not at all. This planaria, then what is its method of division then? What is the method of division or reproduction for planaria? Planaria shows transverse binary fission otherwise. Transverse binary fission is the regular method of reproduction for the planaria. What is transverse binary fission? The body will just obliquely divide into two. The nucleus divides into two, and two organisms develop. That is their regular method of reproduction. What are we learning here now? Regeneration. Under the concept of regeneration, why do we study planaria then? We study planaria because quite often it is found that the planaria, when it accidentally breaks down its body into few fragments, this can give rise to a new organism. This can develop the upper part and the lower part, and a new organism can come out. This part can develop the lower part of the body and a new organism can develop. So now, when we are talking about this type of a condition, we would say that what would happen to these different fragments, something like this, if I understand that this was the upper part of the body. Now, this was the upper part of the body. What has happened? This upper part of the body very much has developed the lower part. But how? How is it possible? Because this lower part has developed only by the method of cell division. Try understanding what I'm speaking. You get questions on this for 3 marks. Why simple organisms like planaria can completely form a new organism? Had this been for the frog, where the frog has lost a part of its limb, is it possible that the entire limb is again formed? A frog or a lizard has lost one of its hind limbs, say suppose. The lizard has lost one of its hind limb. Possible that the lizard body will again develop the entire hind limb, or the left limb, will it develop the entire lizard back? No. In case of this planaria, what's happening? This part can very well, by simple cell division, form the rest of the body. What about this one? If I talk about this part also, now this is the fragment. I talk about the middle fragment, say suppose. Now this is the middle fragment. Now what will happen to this middle fragment? This middle fragment will very much form the upper part and also the lower part. Yes, and the new organism can be formed. नहीं, ग्रो होता भी है तो बहुत रूडिमेंट्री छोटा सा ही ग्रो होता है, नॉट दैट लॉन्ग। जो नॉर्मल पहले था उतना लंबा ग्रो कभी नहीं होता है। एंड इफ आई टॉक अबाउट द लिम्ब, से सपोज द अपर लिम्ब अगर डिस्ट्रॉय हो जाता है, लोअर लिम्ब अगर डिस्ट्रॉय हो जाता है, we come across even human, no? You also must have come across many human, many individuals who have lost their arms due to an accident, arms, fingers, legs, due to an accident. Does the body is the body capable of reviving and regaining the lost limbs? No, not possible. But to some extent only, some that healing of the skin and the muscles, only that much is possible. Now, what is the reason? यही basic concept पे you get a question. The question is that in simple organisms, a complete new organism can be formed by the method of regeneration. But why not in a complex organism like this, or even human? In case of human also we come across, whenever somebody suffers from any major accident, only the skin heals back, the muscle heals back, that much only. Can the bones be formed again? No, the bones and the skeletal system, nothing can be formed. So यही concept यहाँ पे भी लिखना है. If it is a vertebrate, they are always having well-developed organs and systems. And the organs and systems will only develop from a particular stage of growth. कब से? From the embryonic life. From the embryonic life हमारी organs and systems develop होना शुरू होता है. So at any time of our life, if somebody suffers from an accident and loses the arm, the bones and the cartilages and all the tissues cannot develop now. So that's the reason only skin heal hota hai. That's it. But what about the planaria? Does it have any well-developed tissues and systems and organs? Nothing. So only few, at the most one or two tissues which are there, and just a small group of cells that can easily be made up by the simple process of cell division, that is mitosis. So if I say cell division known as mitosis can only result in the development of few cells and tissues, in the development of few cells and tissues, but cells and tissues can be formed, but can organs and systems be formed? No, organs and systems cannot be formed. So what is the answer that you write? Complex organisms cannot give rise to new organisms by regeneration, but simple organisms can. You have to always answer with this concept that if it is a developed vertebrate complex organism, it is made up of variety of organs, tissues and systems. And these tissues, organs and systems will always start its development always from the embryonic life, always from the embryo. From the embryonic stages the growth will start and get over in a period of time. तो यहाँ पे तो कोई organ system नहीं है, so that's why it is possible. It's a very simple organism with consisting of only simple few tissues and cells. So by simple cell division, this can be revived back. Complex organisms in the lizards and the frogs and even in human, this cannot be revived back. We talk about human. Does regeneration happen in human? Yes. Regeneration happens in human. You talk about the liver. Liver is an organ which also shows regeneration. But the moment you talk about bones and cartilages, no, it doesn't show regeneration. Only the surrounding skin and muscle will heal. That is it. And the bones and the cartilages, nothing will be formed there. Clear? This concept is clear? So is regeneration a method of reproduction? Is regeneration a method of reproduction? No. Regeneration is never a method of reproduction. It is only a method to replace the lost body parts of an organism. यहाँ पे भी lost body parts ही हो रहा है, but since they don't have any tissues and organs, वो lost body parts के process में organism full develop भी हो जा रहा है. Are you understanding? क्योंकि इसके body में तो कोई tissue organ system नहीं है, few cells ही था, जो by cell division repair हो गया. But I talk about a human. Again, I repeat this example. I talk about a human who has lost a finger. क्या हो रहा है? Cell division हो रहा है यहाँ पे? Yes. Cell division होके skin heal हो रहा है. Cell division होके muscle heal हो रहा है. But formation of bones, cartilages, etc., nothing is happening. So can regeneration help in the formation of complex tissues and organ systems? No. Regeneration can only help in the formation of few cells and tissues. That's it. Okay? So here है the concept of regeneration, and this is, remember, the main methods of asexual reproduction that we are studying is this, and this is an exceptional method that we have come across. Next is, we talk about vegetative propagation. After this, we talk about vegetative propagation. Now, what is vegetative propagation? After the concept of asexual reproduction, we have to understand what is vegetative propagation and what are the methods. To understand the concept of vegetative propagation, we first of all have to know what is the method, what is the meaning of vegetative parts. When I am talking about the concept of vegetative propagation, first of all, to understand what is the meaning of vegetative parts. Vegetative means non-reproductive parts. For a plant, when we are talking about the non-reproductive parts of the plant, which are the non-reproductive parts of the plant? The non-reproductive parts of the plant would be the roots, would be the stem, would be the leaves, would be the buds, etc. Non-reproductive. The reproductive parts कौन सा है? Flower. Flower is the reproductive part. So let us talk about some methods which come under the concept of vegetative propagation. We are talking about vegetative propagation relating to the roots and the stem and the leaf. The leaf already I have mentioned. What is the process for the leaf already we have discussed? Budding. Leaf already I have discussed. Budding. Which was an example, the type of the plant which shows vegetative propagation? So under the concept of asexual, already I have taken that. Budding, the method is known as budding. So that is for the leaf here. Now let us talk about methods of vegetative propagation. The first is cutting. Very simple a method. We are talking about any type of a plant. Say suppose we talk about any type of a plant, and if it has to be a method known as cutting, what has to be done? A specific length of the stem has to be cut. A specific length of the stem has to be cut. Now, this particular length of the stem that we understand should be something like this. The stem cutting should be something like this. The stem cutting cannot be this way. This is wrong. The stem cutting cannot be this way. Why the stem cutting? If at all you have come across any gardener, if at all you have seen a gardener putting the stem, you always see that the gardener would always slash the stem obliquely. And जो stem cutting है वो लगभग इस तरह का होना चाहिए. Can you tell me why? And now that I try to put them in the soil, after a particular period of time, what is expected? Some fine rootlets should be developing. Some fine rootlets should be developing, cell division hoga, and this will be healed up, and then the stem, and after some time we get to see a fresh leaf coming up. So ये जो process है, this particular process is known as cutting. What is the significance? ये जो cut है, why the cut has to be oblique? Why the cut has to be oblique? This is one precaution. You are asked in the paper that in the entire process of cutting, what are some precautions? What are some specific methods to follow so that we get a greater amount of plants in a short period of time? Because vegetative propagation इसीलिए किया जा रहा है so that the farmer can get large number of plants in a short period of time. The farmer is not wanting to wait for the flowers, pollination hoga, fertilization hoga, and everything is a matter of chance. So the vegetative propagation is a method which is a comparatively a surer method. Let us try understanding what are some of the benefits of vegetative propagation. Benefits of vegetative propagation is it is a surer method. Meaning we are straight away talking about growing of plants from the stem and the leaf and the root. यहाँ पे pollination है, यहाँ पे dependent on any other agents है. Is there any factor on which the processes depend on, like the wind and the water and the atmosphere, etc.? Nothing. Is the process again dependent on so much of nutrients and fertilization and gametes? Nothing. So this is comparatively a surer method. Now, after a particular period of time, if this particular process is successful from one particular plant, we may get easily 15-20 stem cuttings, 15-20 root cuttings हो सकता है from one well-developed plant. 15-20 stem cuttings, 15-20 root cuttings easily हो सकता है, and that many number of plants. So can we write vegetative propagation also gives large number of plants? What is the other benefit? Large number of plants in a short period of time. Large number of plants in a short period of time. So this is another benefit of vegetative propagation. One is it's a surer method, not dependent on any particular agents, factors. Okay, ये हो गया cutting. Let us talk about the second method. The second method is layering. What is layering? Layering is generally done or takes place in plants which are comparatively with a softer stem. Plants जो shrubs हो. For example, layering I would say examples like the grapes, examples like the jasmine, example like the coriander. Now, in this different type of plants, what is happening? It is easier to manipulate, even for water hyacinth and all these plants also, mangroves also, grasses. If I talk about layering, even for grasses, all these. Now, what is the natural method? Let us talk about two types of layering. Now, say suppose I talk about one plant here. This is one particular type of a plant I'm talking about, maybe the grapes or the jasmine. In layering, what happens? These are the roots. From these roots, some extra additional roots known as adventitious roots develop. And that always develops a majority of the plants. So, इसमें क्या हो रहा है? This particular plant giving rise to root systems, and the roots now developing above the soil. The roots have started developing, coming above the soil. The roots have now started coming above the soil, and again from here, another set of rootlets develop. From here, as the roots are all running, now secondary roots running above the soil. What are we naming these type of roots as? इसको बोलते हैं adventitious. These are known as adventitious roots. These are the secondary roots. Developing over the soil, spread up over the soil, and then start developing. And after a particular period of time, again, another new set of plants developing from here. Is this process very fast? Yes, very fast, rapid. In a very short period of time, large number of plants can be formed. So, can I also say that in this particular method, one more feature is all the plants that we get are identical. Yes. The moment I talk about vegetative propagation, why is this method so much preferred by the agriculturists, farmers? Because identical varieties they can get in large numbers in a short period of time. Good varieties of plants they want to get in a short period of time, large numbers, and they would generally go for this method. So this is a method where plants can be grown with the help of the stem, plants can be grown from the root. Now this, what I showed you is a natural method of layering. Layering can be of two types. ये है natural method. But artificial method is also there. What is the method? Artificial method means human manipulation है थोड़ा सा. So what is the concept of artificial method? Say suppose now this is the grape or the jasmine, and the farmer wants large number of plants in a short period of time. So what he would do is, these are the branches. This is the particular plant, and this particular plant is giving rise to branches and then the leaves. So what would the farmer do is, he will try these branches which are developing, he will try to bend these branches and put them into the soil. Cover them with a heap of soil here. What is he doing? What is this part? This is nothing but the branch. This is the branch of the plant. In this particular method, can we think of a plant with a thick stem? No, we cannot think of a plant with a thick stem. I talk about this is the plant. Any of these to remember, the jasmine, the strawberry, the grapes, etc. Now here, if these are the branches, and the branches are giving rise to leaves, etc. What will it be done? The branches, since they are softer, immediately they will not break because these are generally shrubs. So what will happen? These branches will be now directed towards the soil. They will be directed towards the soil and immediately they will be supported with a stick tied with a rope and now covered with a small extra amount of soil here. This is what is known as artificial layering, mound layering both this. So this is what is known as artificial. So layering can be natural, layering is majorly artificially also done. After a week time, what will happen? We come across that ये जो part of the stem था, this has started drying. ये dry हो जाएगा after some 3-4 weeks. ये dry हो जाएगा. Majorly we are now dependent on this part. So धीरे धीरे what happens? We remove this stick also. We take out this stick and we get to see that no, this is already established in the soil. जो branch which was dug deep inside the soil and supported, it has established now in the soil. Roots have come up and a new plant is now developing from here. If this is the method which has been done manually, this is what is known as artificial layering. Which are some plants? These are some plants which are rows also done by this method. So this is what is known as natural or artificial layering. What is the benefit? Benefit क्या है? It is a surer method. Large number of identical plants can be obtained. And the farmer generally does not have to wait for any factors and pollination and etc. etc. So there is no matter of chance. ज़्यादा से ज़्यादा plants बनेगा, that is more or less sure. But now there is one method we are going to talk about, another concept of vegetative propagation, and that is grafting. Now the grafting concept is a bit different. I'm talking about cutting and layering, but now that I talk about grafting, grafting is a different method. Here what's happening, I may talk about two different varieties of well-developed plants. Maybe mangoes, maybe apple, grapes, guava, etc. etc. Now what is happening? In this method, when we are talking about the concept of grafting, why is grafting done? Grafting is done to obtain improved varieties. Grafting is always done to obtain improved varieties of the plant. Grafting is not done to get identical varieties. That's the difference between these two and this method. What is done in grafting? We would generally have to select what? This chapter must be done in school, no now? Over in school? तो क्या है? What is the basic concept of grafting? Tell me. You tell me first. कितना समझ में आया बताओ? Done in school, no? This is over? Not yet? बताओ. Something you should be knowing, no?
Yes, so vegetative propagation में मतलब plants जल्दी grow जाते हैं short period of time में farming को.
तो ये question आएगा तो नहीं लिखना है.
एग्जाम में ऐसे सिमिलर क्वेश्चन आए तो कुछ आएगा ही नहीं था बिकॉज़ हमने तो सीधे में पढ़ाई नहीं है.
Layering, you will be getting this type of figures in the paper. Figures आएंगे question में, case studies आता है question में, where you are asked cutting का basic इतना समझ में ही आना चाहिए. What is cutting? What are the benefits of cutting? Cutting और layering के benefits common है. ये पढ़ना ही है. In the volume also we have given that. So volume में भी the three methods are there. I want all of you to write down vegetative propagation के significance है भी volume में, टिक करके रखो. You'll have to read all those significance for 2 marks, 3 marks as it is आता है, and differences. What is the significance? Layering, only artificial layering is not there in detail mentioned, but this I am comparing and I am telling you this. Natural method of layering, cutting, you have to know definitely. And now grafting. Grafting is, though it is a method of vegetative propagation, it is not done for getting identical varieties. It is always done for getting better varieties, improved varieties. Now, better varieties ka matlab, it has to be two similar plants, almost of the same species. It has to be two similar plants. Say suppose I talk about this is one type of a mango. Try understanding. This is one type of a mango, I may say, and this is another type of a mango. What is happening? Why grafting? Grafting here is because I may come across that in this first species, the fruit is not that nutritive. The fruit is not nutritive, but the plant is resistant to diseases. Now, this is an example with which you have to understand what is the meaning. So, I'm saying the plant is resistant to disease. What is the meaning of resistant? Does not easily get infected by the pathogens. So, this is true for always plants. I'm just picking up two characteristics to understand. This particular variety of the mango does not give rise to nutritive fruits. Ye एक undesirable characteristic है for the plant. But what is the good characteristic? Resistant to diseases. But the other side, here the fruit is nutritive. The fruit is very much nutritive. And this is sensitive to pathogens. Now, there are many characteristics. Last year, we have also studied one full chapter on this. Don't you remember? Improvement in food resource, wahan pe bhi lagbhag some concepts we had studied there. Now try understanding what is the concept of grafting in this. This particular plant has one or two good characteristics and one not a very desirable characteristic. Now the farmer would try to remove this characteristic, make a type of a variety where this and this comes together. In the entire process of farming, agriculture, plants, farmers always try making varieties which has got more and more of good characters and then go for any of this method possible to get identical varieties. Now what they will do, they will try selecting one particular plant which will remain attached to the soil. What will this be termed as? This is known as the stalk. What is it? The main plant which has got majority of good characteristics attached to the soil. Then cut out a small portion of this and try putting it here. But in this selection, the two plants should be more or less of the same species, related species. We cannot choose abruptly any two plants. So now these two plants come here. And what is this known as? This is known as the shock. Now what? A large number of tissue tapes. What is to be added here? Disinfectants should be there. Can you tell me why disinfectants? To avoid any type of bacterial infection. Plants also are affected by bacterial viral infection and a large number of hormones. Which hormones? To initiate the process of cell division and growth faster. Which hormones? Cytokinin, to allow the process of cell division very fast. So now this will be done here. A large number of hormones, disinfectants are applied there. और दो जो stem आएगा, both the stems are tied so that they don't fall off. And in a period of some 3-4 weeks, it is found that the stem, both of them have more or less established above each other. If all the conditions are favorable. Soil, well water, temperature suitable, everything agar theek hai, then in 3 to 4 weeks time, both the stem get attached. Matlab cell fusion already ho gaya hai. And after that, this will just dry off and fall off. And this is now specific plant. This is a improved variety, and this is a new plant that we come across. And in short period of time, we get to see now leaves, flowers developing. तो ये जो मेथड था grafting, are we talking about formation of identical varieties in large numbers? No, we are talking about formation of better varieties, improved varieties. So what is the significance of grafting in the volume? The points are mentioned. Please look into that page and tick karo. Generally with tapes, tissue tapes आता है, or ropes. Generally farmers would use ropes also. या तो when it is done in the laboratory, also in the laboratory conditions में जब किया जाता है, there are various different types of tissue tapes आते हैं, which automatically they will dissolve also. Otherwise with ropes also there, huh? Yes. So there are various different this latex tapes also are there, which dissolve हो जाता है अपने आप, and they will. And now in a period of 3 to 4 weeks time, if conditions अगर सुन now I'm just telling it so simple. सुनने में बहुत simple लग रहा है कि हाँ ये हो जाता है, and then no, quite a number of times they don't even get attached. मतलब self fusion नहीं होता है, and again they will try. So if this works out, this is a better method of making improved varieties. So what is it? Here we have talked about vegetative propagation. We have talked about asexual modes of reproduction. Regeneration was one exception, remember. Clear है? ये सारे topics clear है?
Both of the bad qualities come up with that.
हाँ आता है. Quite a number of times that also happens. So this is just trial and error method. Quite a number of times it is found that the flowers which are developing, fruits which are developing are not of the desired variety. So now that's discarded. Again they start fresh. Again they start new. So this is just an experiment. Most of the time they'll face failure. Again they come out successful that way. So clear है? Untick those points from the volume. This part straight away points लिखना होता है. You will get 2 mark, 3 mark and case study, and you should be able to answer questions specifically. Next is we try now understanding about the concept of sexual reproduction. The concept of sexual reproduction. Now, the moment we start talking about sexual reproduction, the first thing that comes up is the words reproductive organs and gametes. Gametes. So here we'll first of all talk about the plant. So I would say reproductive organs. Since we are talking about the plants first, the reproductive organs, which are the reproductive organs? The reproductive organs would be the flower. Define the flower. Quite a number of times you get this question 1 mark. Define the flower. The flower is a bright colored structure of the plant which has got different type of fragrance. All these things you are not supposed to write. Nobody is bothered about the fragrance of the flower. Eh? You are only to write down what is a flower, define a flower. What are you to write? It is the reproductive shoot. What is a flower? It is a reproductive shoot which contains the male and the female reproductive parts. So we are going to start with this. Write down. Define a flower. Sexual reproduction in plants, right? Sexual reproduction in plants. Under that you write, define a flower. लिखो fast लिखो. अरे speed up करो. पेन निकालने में 4 सेकंड लगेगा. Define a flower. What are you supposed to write? Flower is a modified shoot. Flower is a modified shoot that contains. Flower is a modified shoot that contains the male and the female reproductive parts. That contains male and female reproductive parts. Take out your pencils and very fast you will draw this diagram. We are going to learn the method of the parts of the flower, the functions, and the method of formation of a new plant from a flower itself. That is what I am asking. fast. This diagram is important. You will get this diagram just for 2 marks to draw or to explain the structure of the section of a bisexual flower. With the help of a bisexual flower, with the help of the section of a flower, explain such and such parts. So either the entire thing, entire diagram with explanation comes for 3 marks, or only the diagram comes for 2 marks, along with other questions. So please take a note, this diagram, correct and rough drawing, meaning the parts has to be correctly mentioned. There is no need of any color sketch, anything. Don't use a pen to draw the diagram ever. Most of you are still, you have already made it a habit. Diagram bolne ke saath saath you take out your pen. See that it does not happen during the exam, otherwise you get nothing there. Diagram with the pen is not allowed. You are supposed to use only a dark pencil to draw the diagram. No sketch, no color, no shading, nothing. Remember, follow proper instructions, otherwise you will lose marks. Complete this diagram, and after the break, we will start understanding what are the parts, functions. Functions, एक part का function याद रखना है, and a process known as double fertilization that gives rise to the seed and the plant. That is what we have to learn from this. And then we'll start with the process of reproduction in developed organisms. Human as a representative we are learning. So this is one for the flower, then for the amoeba, and then for the human. Only these 3 examples we have to study. Take a break and we'll again join class.
Guys, please watch Kera and Patrick.
Anitra